and I'm going to present you my research work made in Faculty of Pharmacy in Lisbon about the development and characterization of ultradeformable vesicles with applications for prevention or even treatment of skin disorders. So starting with the summary of my presentation, I'm going to introduce you these ultradeformable vesicles, or just UDV, and then I will explain how to prepare and characterize those UDVs, including topical delivery studies, and finally I will give you some examples of therapeutic and dermocosmetic applications of these UDV formulations. So starting with some theoretical aspects, so as you all know, the skin is the largest organ of the human body where the drug can be easily applied, but the drug must penetrate through different skin layers and reach the target for topical delivery or even the blood circulation for transdermal delivery. But the problem is always the low diffusion rate of drugs across the stratum corneum, the main skin barrier. So, how to solve this problem? Perhaps with physical methods, as we have seen this morning, and with nanotechnology, including lipid vesicles. And the first lipid vesicles to appear were the liposomes. However, due to their poor skin permeability, liposomes are only suitable for topical drug delivery. So UDV appeared as an improved and innovative tool for dermal and transdermal therapies, with numerous advantages over these conventional liposomes, including a high patient's compliance and also a very easy production, as you will see. I will present you 3 UDVs: transfersomes, ethosomes, and transethosomes. Starting with transfersomes. Those were developed by Zeutz in Germany in 1992, and they are composed by a lipid bilayer and an edge activator, which could be one of these surfactants here, and also by an external and internal water phase. Now there are other generations of transfersomes with some modifications in their composition. Regarding the mechanism of skin penetration of transfersomes, once applied on skin surface under non-occlusive conditions, it will start to occur the excess water evaporation from this formulation, and this will establish the hydrotaxis or natural osmotic gradient through the skin, and this force will push these vesicles into skin layers. On the other hand, this skin penetration is also facilitated by the vesicles' deformability due to the action of surfactants in their composition. So that's how we can have a dermal or even a systemical delivery. Now, regarding ethosomes, those were developed by Toito in Israel, and their composition is quite similar, a lipid bilayer and a water phase and ethanol instead of surfactant. Regarding the mechanism of skin penetration of ethosomes, this could be justified according to the skin penetration properties of ethanol, because this alcohol will interact with the lipid bilayer of stratum corneum by disrupting the organization of the lipids and decreasing their melting point, which will consequently increase the fluidity and cell membrane permeability. So that's how ethosomes could penetrate and deliver the loaded drug into deeper skin layers. And this film represents exactly this skin penetration mechanism. Here we can observe that the vesicles will penetrate into deeper skin layers in its intact form. You see, it will be in its intact form due to its high elasticity. And finally, transethosomes were developed by Song, and they have both ethanol and an edge activator in their composition. So perhaps these most recent vesicles will have the advantages of both transfersomes and ethosomes. So, The aim of this work was to compare the skin release and penetration profiles between these three related UDV in different situations, with a higher and a lower incorporation efficiency, which could be easily obtained by entrapping a lipophilic and hydrophilic actives, respectively. And the models used for this were vitamin E and caffeine. In fact, this last case, the lower incorporation efficiency, here we also intend to better observe the role of the nanocarrier itself regarding the skin penetration, as I will explain you. Starting with the methods of this work. Here it is the formulation of these three preparations. We had just 1% of the active, 10% of the lipid, which was soybean phosphatidylcholine, SPC. We tested two different surfactants, 30% of ethanol in ethosomes and transethosomes, and purified water as the main vehicle. Starting with the preparation of transfersomes, quite easy. We just add all these components, and this mixture was stirred for at least 1 week, just that. Ethosomes were prepared according to the classic method, in which the ethanol and lipid mixture is stirred and immersed in a water bath at about 30 degrees. After obtaining the complete dissolution, we had the water drop by drop through a syringe needle that we insert here in the bottle cap. And as we had the water, we could also observe the formulation changing its colors as the vesicles are being obtained. We stir for more 5 minutes, and after that, we just kept this formulation at room temperature. Regarding transethosomes, those were prepared according to these two different methods. Method A, corresponding to transfersomes, and B, to ethosomes preparation. And at the end, all these three formulations were filtered under nitrogen stream using this filtration device that you see here, and using a polycarbonate membrane that we put here in order to obtain that vesicle size. Now, regarding the characterization of these vesicles, we start to observe the morphology of the vesicles by transmission electron microscopy. We measure the size and zeta potential by dynamic light scattering and laser Doppler anemometry, respectively. We calculate the incorporation efficiency and active entrapment yield inside the vesicles after the separation of the incorporated active fraction by ultracentrifugation. Then we assayed the active compound by HPLC system and the lipids using a commercial kit. We have also calculated the flux and elasticity of these vesicles according to these equations, and also using the same filtration device, but now with a smaller pore size membrane. We have measured viscosity, because this was for topical delivery at these conditions. And finally, we have performed the topical delivery studies according to official guidelines. And according to that guidelines, we must use France diffusion cells. So we just applied here in the donor compartment an infinite dose of the formulation and under known occlusive conditions. And we have also ensured that here, down in the receptor phase, we have sink dissolution conditions. Then, at certain periods of time, we collect the receptor phase and assayed by HPLC in order to evaluate the releasing profile, if we put here in the middle a synthetic membrane, or the permeation profile, if instead we put just skin. And finally, we have also evaluated the penetration into different skin layers by the tape stripping. So, the results of this work, starting with the macroscopic appearance. As you can see, transethosomes were the most translucid formulation. And curiously, with electron microscopy, it was observed the same, including the presence of multi-layers and also the spherical form of these vesicles. Now, this huge table is just to show you how important it is in research lab to standardize all procedures, since the formulation, composition, preparation, and even characterization methods, in order to better compare all these related formulations. Starting with the size. Our main goal was to obtain 150 nanometers, and we have obtained that, except for certain formulations of transethosomes, especially those prepared by the transfersome method, perhaps due to a higher steering period and also a better rearrangement of the vesicles components. That's why we decided to use the transethosomes prepared only by ethosomal method for the topical delivery studies. Regarding the polydispersity index, this should be the lowest as possible in order to indicate that we have an homogeneous population. And that's exactly what we have here, except in one formulation, transfersomes with vitamin E, in which it was a little bit higher, perhaps due to the specific location of vitamin E inside the vesicles membrane, and also to its higher incorporation efficiency. Regarding the zeta potential, the charge, it was negative, as expected, attending to the formulation's composition, and the highest values were obtained for sodium cholate formulations, because this is a negative surfactant. And also the higher values indicate a higher stability, considering that there is a higher electrostatic repulsion between vesicles. And in this case, the actives did not influence this parameter. Now, regarding the active entrapment yield, it was higher for vitamin E than caffeine, as expected, attending to the lipophilic nature of this vitamin, which could be easily incorporated in the vesicles membrane. On the contrary, the hydrophilicity of caffeine will difficult its incorporation into the aqueous internal space. And finally, the viscosity was quite suitable for topical delivery, being a little bit higher in the presence of ethanol, because this will fill the voids of the hydrogen bond and then fortifies the structure. And the vesicles flux and elasticity. As you can see here, transethosomes were more deformable than ethosomes than transfersomes for both vitamin E and caffeine formulations, including the empty controls. Finally, the topical delivery studies, starting with the releasing profile with formulations with vitamin E. As you can see here, the order was exactly the same as observed for the flux of these vesicles. Why? Because transethosomes are the most deformable vesicles, so a higher membrane passage and active release would be expected. However, the same did not happen with formulations with caffeine. In this case, the releasing order was exactly the opposite, probably because this is more related with caffeine solubility, which was higher in transfersomes, and thermodynamic activity rather than in UDV deformability. And it was also observed a higher release for solution controls of each UDV. In this case, we have also tested the solution controls attending to the low incorporation efficiency of this active. And the skin penetration here, the first graph is in the stratum corneum, and the second one in epidermis and dermis. So as you see, transethosomes contributed to the highest amount of the skin penetration, as expected, attending to the highest releasing and deformability. Again, the same did not happen with UDV with caffeine. In this case, we have also observed a higher skin penetration of those solution controls. So here you can see we proved that these nanocarriers did not improve the passage of the known incorporated fraction of caffeine through the opening transitory skin pores, as the UDV passed through the skin, compared to the solution controls. Perhaps due to the absence of lipids in that solution, and lower viscosity and higher caffeine solubilization. Now, the skin penetration of these UDV were also confirmed by fluorescent confocal microscopy. And in this case, we incorporate some dyes in this UDV. So as you see, we have obtained a successful UDV penetration into different skin layers here, and also the cellular uptake. And these vesicles were localized around the nucleus of keratinocytes. Now, regarding the therapeutic applications, here I represented the in vivo studies of tretinoin and lycopene-loaded UDV. According to this histopathology of mice skin treated with tretinoin-loaded UDV, the irritation potential of this formulation was much lower than the commercial formulation. You see here some signs of skin inflammation, which were not present in tretinoin-loaded UDV. And the therapeutic effect of the drug was still maintained, as observed according to epidermis proliferation. And we have also evaluated the therapeutic effect of lycopene-loaded UDV, which was in this case demonstrated by the inhibition of edema formation caused by antraline. And besides this inhibition, it was also inhibited the sebaceous gland's hyperplasia. Curiously, that hyperplasia was still here after the corticosteroid treatment. So a very nice result with lycopene and also tretinoin in these UDV formulations. Now, regarding dermocosmetic applications, nowadays there are several companies that are investing in nanotechnology, especially L'Oreal. And why? Because these nanocarriers, including lipid vesicles, have different functions in formulations. Besides being nanocarriers, they can also stabilize these formulations. So we can have several cosmetic and dermatological applications depending on the actives that are incorporated in these vesicles. Most of products in market are liposomes, but there are also now new products with ethosomes and transfersomes. And finally, this last slide is just to show you the huge number of nanomaterials that are used in cosmetic products, including cyclodextrins, solid lipid nanoparticles, crystals, among others, besides lipid vesicles. So the conclusions are UDV are useful as topical and transdermal delivery systems, besides contributing to improve technological drawbacks and increase the therapeutic index, as we have seen for tretinoin and lycopene. And they are also useful for dermocosmetic applications. Among them, transethosomes are the most deformable vesicles, perhaps due to the synergistic combination of ethanol and surfactant in their composition. So they are suitable for a deeper skin penetration, but only with a high incorporation efficiency, as we have seen for vitamin E. So the future perspectives of this field, I think it will pass through hybrid and synergistic systems in which we combine them to achieve several applications. Also, a new generation of textiles which incorporate these nanocarriers, and also the expansion of the study of new nanomaterials to reach the market stage, and finally, update, and this is very, very important, to complete the regulatory issues, mainly nanotoxicology issues. This is very, very important. So thank you very much for your kind attention.